Okay, so today, so here's the plan for today. So I'm going to follow up or continue things I've been telling you about motif and modules the other day. So I will tell you about, so these little names here are standard names for modules that are important and that have been discovered in a very similar way than the modules I've already described, the self-regulating gene. And these guys are what you see. They display richer dynamics and in some way are more generic. I think that even more generic than what I showed you yesterday. That was yesterday. Then I tell you a word about synthetic biology because there is a connection between this idea that you have motifs in gene networks that appear in evolution and what people tried to design actually in the lab. And I think it's interesting to actually compare these things and see how they differ and how the difference in the commonalities. If I have time, I will tell you a word in the end about modularity in the gene networks and hypothesis on how it evolved. And all of this will be a kind of a very big introduction for what I will tell you about tomorrow, which is really more focused on my work, actually my own work, which is the idea of how do you simulate network evolution, essentially, while keeping biological function in mind. Okay, so let's start with part one, which is the study or the introduction of some interesting motifs. So the first motif I want to tell you about, so it's actually a very important motif. This is called the feed-forward loop. And I think Paul yesterday used this word of feed-forward to describe one of the modules. I think yesterday's talk was quite interesting to show you how this kind of small modules, small networks can really have functional roles and can also evolve and be modified by evolution. I think that gave you a sense of why it's important to focus on these little motifs. So feed-forward loop. So these motifs were also discovered in the very same paper I showed you yesterday, where they compared again a randomized network with the actual network, keeping the connections, so the number of connections and number of nodes constant. And so feed-forward motifs. So I have a couple of figures, like in terms of quantitatively. So so let me just draw first this module. So this is a module made of 3 genes. So let's call it an X, Y, Z. Yeah, and so of course we're now moving up to modules with more genes. And so the connections are like this. And I'm going to put a circle here. And I tell you why I put a circle, a little circle here. So this is the idea that you have an input X, which is like it's kind of a cascade. It's upstream in the cascade, and it directly regulates something, the most downstream gene of the cascade. And then there is an intermediate step, another intermediate parallel step between X and Z. So that sounds like something very simple. Now the reason why I put these little circles is that this guy here, it can be either positive or negative. And by this, I mean, yesterday I told you about the self-regulating gene that could be either self-activating or self-repressing. This is what I mean here. It could be either an activation. So again, my convention that activation will be simply an arrow like this, or a repression, these T arrows. Okay? And so now what you can do is that you can characterize the feed-forward loops. And so there's a bit of nomenclature about how you characterize them. So everything, not everything, but this has been done in a paper, again, by the group of Alon, so Mangan and Alon, in a PNAS paper in 2003. And they list all the possibilities. So basically, this guy can be positive or negative. There can be positive or negative, and this guy can be positive or negative. So you see, it gives you some numbers of feed-forward loops. And so the two most interesting I want to tell you about today are actually I'm going to tell you about 3 different ones. Yeah, 3 or 2. That's 3. Let's do 3. So the first one I'm going to tell you about today is this feed-forward loop. So essentially everything positive. Okay, and so what you do usually when you study feed-forward loops, that you study how contradictory are the two branches. So if you have both, like, so here you see it's a positive interaction on this branch globally and a positive interaction also on this branch globally. So you could have a coherent positive feed-forward loop. So coherent because it's the same on both sides. It's both activating. So this guy, I have some numbers. So this guy, you found 30 of them in the E. coli transcriptional networks that you saw yesterday. And you also have, these people have done it also on yeast transcriptional network, and you also find 30 of them in the yeast transcriptional network, similarly. Okay, so that's a coherent positive feed-forward loop. And so another one I'm going to tell you about is, now this is called an incoherent feed-forward loop, because now we have a different relation between the two branches. So the difference that you flip this guy from positive to negative. So this is an incoherent feed-forward loop. And okay, I dropped the feed-forward loop. Yeah, so now I write it on FFL. And so this guy here, you have 5 in E. coli, and then 21 in yeast. Okay. And the last one, so I'm going to say a word on each of these modules, feed-forward loop module. The last one I want to tell you about, I'm writing down immediately. So this is another kind of incoherent feed-forward loop. Now I'm going to put the negative relation here and the positive relation here. So that's another kind of incoherent feed-forward loop. And I did not write down how many of them there are in E. coli and in yeast, but I'm going to use it as an example of the diversity of behavior that you can generate with these networks. So okay, so again, the idea is that these networks are overrepresented in actual transcriptional networks. And so these motifs, I should not say these networks, the small motifs are overrepresented. And so the idea is that if they're there, it's because they do something that must be interesting for the cell. And as I showed yesterday, this something could be things like bistability or things like interesting kinetics or stuff like that. And so it's very natural to try to study a priori what these networks can do, and then try to match to actual experimental observation. And that's what we're going to do now. So I'm going to start with this guy, the first one. And so this is again an observation made by these people, Alon. So the idea, so there is one little catch that you need to deal with now, is that, so when you say X activate Y, it's pretty straightforward. I write an equation, I say X dot is equals to some function, so Y dot is equal to some function of X minus X. It's pretty straightforward. Now you need to tell what happens when you have two arrows pointing to the same genes. What do I mean when I have two arrows pointing to the same gene? Is it some kind of N function or function, or what does that really mean? And so, of course, this depends a lot on the biological problem you're going to consider. In some cases, it means that the two activators need to be present at the same time. In some contexts, it means that it's either or to activate Z. And for the feed-forward loop to be actually interesting, I mean, clearly, if this is more like an or function, I mean, you don't really see what is the difference between these two guys. So the most interesting behavior for this guy is obtained if you need to have these two branches essentially activated at the same time. So we're going to study this network where implicitly we basically add a kind of Boolean gate. Let's say it's kind of a Boolean AND gate. Okay, so that you need to have essentially. So what that means here is that you're going to consider a network where to activate Z, you need to have both Y and X to be activated at the same time. And the only addition compared to a Boolean, traditional Boolean gate would be that now we assume that also that X activates Y. So this simple network has been proposed to act as a so-called persistence detector. Okay, so what does that mean? I'm not going to write equation. I'm going to give you a very, very intuitive feeling of, and of course, everything I tell you, you can put it in equation. That's really not difficult. Let's start by assuming that as a function of time, let's try to see, intuitively what happens. Imagine that you have a signal X, so maybe I'm going to use colors now. Okay, I just need 3 colors. Okay, so let's say I activate X is turning on, so something else is turning on X. Okay, and then let's say something else is removing X. So this is my X. Okay, and so now let's assume that the thing, all the kinetics is relatively fast, so it's pretty straightforward to see what will happen. So X is going to activate Y, so Y is going to turn on after a delay because it takes some time for Y to activate X, and then for the same reason, Y is going to die after a delay. It needs to be degraded. Yeah, it's probably more like exponential decay, but you get the idea. Okay? And now what will happen? Well, if you say there is an AND gate for Z, well, it means that this Z is activated only when X and Y are present. So you see, what Z is going to behave like that. Okay, so it's going to do this. Okay, so that's Z. Z is yellow. Z is yellow, Y is green, X is blue. So it's not too exciting at this stage. Just X activates Y, and X and Y activate Z. That's really very straightforward. But now what is interesting, and what was proposed by Alon and co-workers, is that now look at what happens if instead of having a long expression of X, I have a relatively short expression of X. So let's see what happens. And trying to have the same, to match the time scale. So you activate X. And now I'm going to say, well, no, X is activated, but it dies relatively quickly. So something like that. Okay, and so now what will happen is that if the time scale where X is activated is yes? Yeah, that would be the protein expression level. And again, I could write an equations, but I don't want to, it's no use writing complicated equations. It's very intuitive. So you put more X, and so imagine that the time scale, the duration for which X is active here is comparable to the time scale of accumulation of Y. So then what will happen is that Y will get activated, but only once X is essentially dying off. So Y is going to be like this, and then, okay, now at this point, X is removed, so Y is going to die similarly, and so you get this, Y is like that. And so now, of course, if you have a situation like this, and if I have an AND gate between Y and X, where Z here, Z, you might have a little bit of Z here, but it's going to die immediately because X is removed. Okay? Okay, so then what you see is with this network then is that so if you say that X is an input and Z is an output of your system, you see that Z is going to be activated only if X is activity for a sufficiently long time in the system. So that's why we call that a persistence detector. Z is active only when X persists. Okay, and so you see that a network like that, again, it's basically performing a computation. It computes a time scale. Essentially, it's able to tell you, how long do you activate a signal, and with a simple network, the simple dynamics of the network, you get a relatively, sophisticated computation based on the duration of the signal of X. So, yeah. Interestingly enough, this design was actually rediscovered. So presumably it was evolved in in E. coli transcriptional network. And I have to say, these people that actually try to look at a couple of these feed-forward loop networks and see if they were really acting like, really behaving like the persistence detector. And they found some examples where they could really show that indeed you needed a sustained signal of X to get a sustained signal of Z. So that they really checked that at least functionally these things were behaving like what they were predicting. And so it turns out also that this kind of design are actually very well known by engineers. They know, and so I don't know if you have. So now, I have a smartphone, but when I was young, I had the dumb phone, and then I don't know how it works now, but your phone. But when to turn this on, to turn the old phone on, you need to press the on button for a long time before it turns on. And so the electric circuits in these little phones were actually really implementing that. So you needed to you need to stay on for a while to turn on the phone. And of course the idea is that you don't want to by accident turn on your phone, And so you really need to want to turn on your phone to actually turn it on. Otherwise you have accident pocket calls and stuff like that. So anyway, that's the first possible function, possible behavior of this little motif. Okay, so now I'm going to focus. Yeah. Yeah, yeah. So the question is, how did they find that you had these feed-forward loops in the E. coli transcriptional network?
Right. My question comes from because later you said that it's like they first knew they are a feed-forward loop, and then they test.
Yeah. Okay. So let me start from the beginning and give you a general perspective of what this is. So what they did, they had these transcriptional networks of E. coli or yeast. Okay, so then what they did, as I explained yesterday, they randomized the connection and try to see how many, what kind of motifs do you see that are overrepresented in the real network compared to the randomized network. So this is a way to test, and the idea is that if these motifs are overrepresented in the real network, they must have been selected by evolution or must have an interesting function. Okay. So that's how they found these feed-forward loops. They identify that these feed-forward loops are part of the motifs that are very present in the actual networks, and statistically they should not be as present as they are in the real networks. And so that's why they say, well, if these networks are present, they must have a function, an interesting function biological, and then this is where theory kicks in, and you say, well, let me study this network, let me study the simple models of the dynamics of the networks, and let's try to figure out what they actually can do. So then my claim here is that these networks, they can do this, they can do the persistence detector. And now once you have made a theory of what they can do, you can go back to the actual network and check if they behave like you were predicting they were behaving. So you can look at the actual transcriptional network in E. coli, identify one or two of these feed-forward loops, and see if you can, so you put gene names, actual gene names here, and then you turn on this gene here and see if it behaves as a persistence detector. And so that's so they've done all this process. Of course, it's a long-term process. It's something that took them 10 years. But that's what they did. Yes?
They first found it's a feed-forward loop.
Yeah.
And then they look if in the natural system, the system is in this set of parameters where you see these dynamics.
Yeah, exactly. So they've done that. But of course, they have not checked all feed-forward loops because that would be impossible. They picked a couple of them. And so, of course, then you can wonder, maybe they cherry-picked the one they found. I'm not going to comment on this because I don't know. But at least they try to check the hypothesis and could check it on some networks. Okay? Yes?
So Z is active only when X persists, or both X and Y persist, or like X and Y are both required, right, for Z?
So x, yes, in this is what the end means. It means that for Z to be active, X and Y should be present at the same time.
So why you written Z is active only when X persists? Won't it be like when both X and Y persist?
Yeah, but okay, so I'm not doing it purely Boolean. I say, there is a bit of delay, you activate, and then you decay, so you don't kill it immediately. If you were to simulate an actual gene networks with Hill function and stuff like that, simulating this, that would look like that. So that's, but of course you could make a very Boolean version where step function and then do this. I just, it's biology, it's fuzzy, that's what this is here. Okay, any other question? Okay, I cannot hear you, sorry.
I was just wondering if it's an OR gate.
Oh yeah, well, if it's an OR gate, it's completely uninteresting because as soon as X is on, Z is on. So Y is not doing anything.
So like that's...
So for this thing to work, it needs to be an AND gate, absolutely. Otherwise, it's completely... And then, actually that's part of the question. Maybe in the real network when they identify modules, but it could be that in many of them, this is an OR gate and not an AND gate. That's why it's also important to check in retrospect. Yes? Yeah.
Is there also a notion of fan in and fan out? Like if a particular output of something is driving a lot of things, so it will, compared to when it's driving fewer things, it'll drive it with high intensity, the fewer things, and with lower intensity, the lot of things.
Okay, no, there's not something like this, but the fan networks are part, the fan motifs are part of the motifs that they have identified as well. So you certainly have fan motifs, but you don't have like distribution of strengths over the motifs because it's more like independent links, except, as far as I know, it's essentially independent. But the fan motif is an important one. I'm not going to tell about the fan, but they also discovered it. Okay? Okay, so now let's move on, and I want to say a word about these two motifs because they prove important, especially in what I discuss tomorrow. So I want to spend some time studying them, and so. Let's start with this one here. So the big difference between the person's detector and this guy is that you turned repression into an activation. And so now when you have a repression, also you need to make an assumption about the, what is the logic here? What is, how do I combine a repression with an activation? And usually a good hypothesis in this context, and that's more or less true biologically speaking if you look at actual examples. It's something I've been very concerned with for the reason you will see tomorrow. The idea is that in most cases the repressor is winning. Like you have a repressor, it basically, it prevents the RNA polymerase to come or anything. So, having it's very difficult essentially to compensate a repression with an activator. It's essentially repressor wins. So with this in mind, you can now study what this thing can behave, and again, it's pretty straightforward and intuitive, and I will show you, especially in a more like in a context where these arrows here have slightly different strengths. And so let me make a simple drawing to explain what I mean by that. Okay, so I'm going to give myself, again, we're using the same convention, I'm going to give myself an axis. And so now my axis, it can be time, but let's assume let's assume it can also be space. So I'm going to call it T, but I could also call it X. And you will have, the network will have a signification in both cases. So X would be space. Okay, and it's probably it should start to remind you of things you've been doing with John Reinitz, especially if I assume, now let's assume that X is, is increasing monotonically. So let's assume that I have X doing this, okay? As a function or time or space. So if I do this, this is now my concentration. So I just assume I take X, and I just assume X is increasing either in time, so it's in one cell it decreases in time, or I assume I have an embryo, and I have my concentration X that is depending on the position of the embryo, like bicoid. You saw bicoid or caudal, this maternal gradient, where the concentration depends on position. Okay. And so now we're going to assume the following. We're going to assume that, okay, so X is going to activate Y and Z, but let's assume that it activates it, in very French flag model way at different levels. So let's say, and so the interesting thing to consider is imagine that this guy is active at low X, and this guy is active at high X. So in other words, you first activate Z, and it's only later on that you activate Y. So now let's see what happens. So I start with Y. So Y is going to be activated only at high concentration of X. So Y is going to look like that. So, and I try to stay consistent with my color scheme. And so let's imagine that Y is like this. Okay, so again, if you remember what John Ryan did, it's very much like bicoid hunchback. You probably remember that hunchback is very well defined above some concentration of bicoid, and Y is really literally hunchback here. Okay. Now, what happens to Z? So if Z is activated, below, at a lower concentration of X, then Z is going to behave like this. Okay, so it's activated first. And then, of course, now what happens is that Y is replacing Z. So now it turns out, but then when Y is turned on, Z is decreasing. Okay. So that's it. And so now what you realize is that this network is basically perfectly implementing a French flag model. Okay, where you clearly have three regions. You have region there is nothing, region where there is Z, and region where there is Y. So this very simple fit for our network, it's basically very convenient to, in a developmental context, to define different regions in the embryo. So it's basically, you could say it's almost like a French flag model network. Okay, and so of course, because of this, if you actually look at, look at actual networks in development, such as what you see in Drosophila, well, you see a lot of these things where, again, I mean, literally you would have bicoid activating different targets at different concentration, and one replaces the other, and that defines different regions in the embryo. So people are actually working in this in a more evolutionary way these days. There are some work where people are building this artificially and then studying how you can make artificial evolution experiments to see, how you can move the boundaries and stuff like that. So this is an active field of research this day to, again, take this network, evolve them, and see how they are, robust to evolution and stuff like that. So that's another example of the fit for network. So as you can see, this network, the minute you start adding more genes and more interaction, it's a bit like the space of possible is starting to expand. This simple network can have very different behaviors, depending on the context, and then you can see why, you can guess why it appears in many contexts, because it's so versatile that it can be used in different ways in different organisms and completely different for completely different functions. Okay, so any question on this? Okay, so now the last behavior I want to tell you about is this guy here, which is slightly different. And I'm going to, now for this one, I'm going to actually write a couple of equations because it's an important network that will come tomorrow, and the dynamics matters for this network. So let me take this network. So I just draw it again here. Okay. Okay, and so to understand, so now I'm going to look at this in a dynamical context. So I'm going to assume that you have a function x of t. Okay, and then I'm going to write down a simple Michaelis-Menten or Hill functions to describe the dynamics of y on z in this context. So z is pretty easy. So y dot, I'm going to assume that there is, so let me write this way. Yeah, let me write down this way. Let's assume that I have a function f of x, and which is like this. So I'm going to put degradation constant. Let's assume I risk, okay, I put a degradation constant. That doesn't matter too much. Okay. So let's assume I have something like this, where f of x is going to be, f of x is an increasing function of x. Okay, and so now I'm going to assume for z, I'm going to assume something a bit like this, where so I need to have both at the same time. So I'm going to say there is y. It could be a function of y, but it's just easier to explain what happens if I assume it's y. And then I'm going to assume, so the thing I'm going to do, I'm going to assume that there is something in the network that is adjusted so that this f of x you have here is exactly the same on this arrow here. So I'm going to say this is y f of x minus some mu z. Okay, so you might find it bizarre that this is the same term, but let's assume it is like this. It's possible to do that, but let's assume for now that this is the exact same term. Okay, so let's look at the network like this. And so what is interesting first to consider is that what happens at steady state. So let's assume that x of t is now, so x a priori depends of time. And now let's assume that x is constant, and let's see what happens. So if x is constant, let's assume x of t is equals to x naught is a constant. Okay, so you just solve the equation. It's completely trivial. So you have y is equal to 1 over lambda f of x0. Okay, and then z, now it's equals to mu, 1 over mu. And so now what do you have? You have y times f of x0. Okay, and so f of x, and then you replace y by its value. So maybe let's call that y0 at steady state. And so y0 here, and then y0 you replace by its value, and then what you have is that you get 1 over lambda mu, and then what happens, the f of x0 disappears. So it's just a constant. So z0 now is 1 over lambda mu, which is a constant. Well, obviously it's a constant, but the important thing is that it's a constant that does not depend on x0. So it's a constant. Okay, so why is it interesting? You know, you get a network, because the network produces a protein Z, and then Z always has the same concentration. You know, that does not seem very impressive. So why is it interesting? It's interesting because this is at steady state. But now what happens if x is changing as a function of time? And let's try to see what happens, and this is where the interesting function is coming. So, and more precisely, I do the following thing. So let's assume, as a function of time, okay, that I start with an x initially like this, and then suddenly I do a step function in x. So x is a bit like it's a bit like an environmental variable. You know, I have, imagine, something like a cell is suddenly changing environment, it's getting, or you increase the salt concentration or the sugar concentration or something like this. This is a context where x, where x is where you get x. Okay, so now what happens to z? Okay, so z initially, it's a constant, so let's assume that z is here, so z is 1 over lambda mu. Okay. Okay, so it's constant initially. Okay, and so now, clearly, at the very end, once x is, if you wait long enough, as I say, you should go back to 1 over lambda mu. and everything is in going back. So at the very end, I have 1 over lambda mu. But what is interesting is that Z is going to change in between. And the reason why it's going to change is precisely because of the presence of Y. So let's think about what Y is doing here. So Y initially is going to be at 1 over lambda f of x0. So maybe I'm going to also write it down. This is Y. Okay, let's assume this is Y in the beginning. Okay, and so now Y, imagine you increase X. So if you increase X, you are at steady state first, Y is at steady state, you increase X. So it means that f of x0 is going to increase. So that means that the production rate of Y is going to decrease. So then what will happen for Y is that it's going to decrease. Okay? It's going to decrease until it reaches new steady state, which is something like, it's going to be a constant. So that's the Y dynamics. And now let's see what happens for X dynamics. So for Z dynamics. So now, imagine just after you change the X concentration, Y, it's still at its steady state concentration, the initial blue line, but then X is increasing. So that means that this term here suddenly is increasing. So since it's increasing, Z is going to shoot up. Okay? So it's going to turn on. And then, well, you know what happens. So the Y is going to decrease. So Y is going to decrease. So as Y is decreasing, this production rate is going to decrease as well. And then you go back and relax to the steady state. Okay. And so now what you see, so that's the dynamics you expect to happen for your system, X, Y, and Z. And then what you see, what is happening here, is that Z, if you look at Z of t, it basically tracks the change of X. It's turning on, it's constant, and then when X is changing, boom, it just spikes and then come back to steady state value. So Z of t is basically a bit like, in some way detecting the derivative of X of t. It's likely only when you change X that is going to signal and then come back to its initial concentration. So this has a name in biology. This is something that is called biochemical, because here's biochemical, but you could have it work in other contexts. Adaptation. So adaptation, this is the idea that you change some input, and then the variable is, there is some deviation in the variable you consider, but then it adapts to come back to the same concentration it had. So it can be used, and it's observed in many contexts in biology. So if you think about, homeostasis, what is homeostasis? It's like you change the concentration of something, and then you come back to the same state. So of course there is always a transition period where the things can you need to signal something, you need to compensate for other variables. But if you want to maintain something at constant level, you need to have some mechanism like this to perform this homeostasis or biochemical adaptation. Another real life example, which you all experience, is that right now we are in a relatively dark room, and so if you look at the opening of our eyes, it's kind of broad, because the idea is that you want to have enough light on our retina to be able to see. But now if I run outside, obviously it's very sunny outside. So in the beginning, when I go outside, it will be a bit, I will have too much light. I won't see very well. And then the opening of your eye is adapting as well, so that you always have the same constant of light on your retina. So in this vision, in this very real life example, X would be outside light. Okay? Y is the opening of your eye. And Z is the light of the retina. Okay? Yes?
Would we also think of this as the mechanism behind immune response? Like, let's say some foreign virus or bacteria comes,
okay,
and this is
about that tomorrow. Okay? A quick answer is yes. But let's talk about that tomorrow. So, but obviously, you know, as you see from these very different examples from real life, and it's not only in biology that you will see that in molecular networks, but you know, these designs, these fit-for networks to adapt to the outside world, this is something that also has been discovered by engineers. And you know, there's many things like control theory. So here, fit-for is not feedback, but you could do something similar with feedback. So there's a huge literature on all of that. But you know, the bottom line is that you realize now that you know these functional approaches of networks are probably very relevant because you see that you know they really correspond to very intuitive things that are happening both in real life and probably at the level of the cell. Okay. Yes.
The profile of that x, so
Yeah.
there is a step function.
Yeah.
So there is the derivative is not defined over there, right? I mean, is that
Yeah,
a problem
well,
in the dynamics, or it's just 1 point so that?
Yeah. Okay. So that's why I put quotation. You know, I put quotations. It's not exactly the derivative, but the idea that you get activated when you have a change. That's a bit an intuitive way to think about it. But obviously, that's not exactly the derivative. That said, if you were to, I don't know, I've done that calculation, but I would be probably close enough to some delta x. Usually, what you find is that delta x over x, it's more like the log, the derivative of the log or something like that in these networks. Which also is interesting because there are things like called Weber's law in a biochemical network where you realize that many of biochemical networks, a bit similar to that, they actually are sensitive to the log and not to the absolute value. So the fact that you find that a network like this is actually computing the derivative of the log is actually quite interesting. So that connection, like there's a whole huge literature on all of this. Like, is it the log? Is it the derivative of the log, or is it the derivative? Things like that. But I'm not diving into that. I just want to give you a very, very simple and crude overview of all of that. Okay. So, wow, time flies. Okay, so feed-forward loop. So I'm done with the feed-forward loop. And so now the last the last module I want to tell you about, this is something called the mixed feedback loop. So I don't think I'll tell you as much on it. So what is a mixed feedback loop? So I mentioned that these networks, feed-forward loop networks, self-activating gene, self-repressing gene, they were discovered by looking at these transcriptional networks and identifying statistically how some motifs were overrepresented. And so this is a discussion we had yesterday. Of course, there is more than transcription in the cell happening. You can have various things. You can have phosphorylation. You can have protein-protein interaction and stuff like that. And so these people, again, this is a group of Alon who really specialized into this in the early 2000s. What they've done is that now, at the same time that you add the list of transcription factors and transcriptional interaction in yeast, say, you also add the list of protein-protein interaction in yeast. Yes?
So I'm sorry to say, maybe go slow. I mean, a bit slow. I mean,
it's too slow?
No, it's too fast.
I'm going too fast?
Yeah.
Okay. Okay, sorry about that. Okay. Okay, so more slowly. So what I was telling is that people looked at the list of transcription factors and transcriptional interactions to reconstruct these big networks, transcriptional networks. But so at the same time, people were doing experiments and they were identifying protein-protein interactions. So like the list of protein interacting. And so now what you can do is that you can cross these two networks, both the transcriptional network and the protein-protein interaction networks, and look at the motifs combining at the same time transcription and protein-protein interaction. Okay? And so then when you do that, you find new motifs, because you find new motifs with actual protein-protein interaction. And the simplest motif that is coming out is this motif called the mixed feedback loop. So MFL stands for mixed feedback loop. Okay, so what does that mean? So using the notation I've been using so far, so the first thing is that you have two proteins, or two genes. It's more like proteins that are important. So X, and you're going to have X that is activating or repressing Y. Again, I'm using the same notation as before. And now, why is it mixed? It's because what you have now is that X and Y, so when you cross it to the interaction, to the list of possible interaction, you get a protein-protein interaction. So I'm going to write it down like this, where X and Y can form a dimer XY. So this is a protein-protein interaction. So the idea, again, is that X, the protein X is going to activate gene Y, but then X and Y can, the proteins can interact in the cytoplasm and form a dimer. Okay? So this is called a mixed feedback loop. And so this motif is overrepresented in biochemical network in yeast. And so, of course, now you can ask again the same question: what could it do? What can it do? What can it do? And so, okay, I should actually give you some references. I think it's important that you see where this comes from. This was done by Jaeger. So this was done by Jaeger, Lotem, and Al. Et al. In PNAS in 2002. 2002 or 4? Okay, I thought it was 2004. Not entirely sure, but. Okay, so now what can you do with this motif? And so actually this is part of my PhD. So I can tell you, summarize in 5 minutes what one chapter of my PhD has been about. And so what is interesting is that this mixed feedback loop, again, it can come in two flavors. Either you have an activator or a repressor. So let's start with the repressor, because that's the easiest case. Okay, so if you have X repressing Y, and then X and Y forming a dimer. Okay, and so what we're going to assume is that X and Y is not able to repress Y. Only the free X can repress Y. So does not repress Y. Okay. So you can write down an equation of that, but the intuition is actually pretty simple. So what is happening if I give you the answer? It's a bistable system. If you choose a parameter correctly, it's a bistable system. And so why is it bistable? Okay, so you can get a simple intuition of that. So if you have much X in the system, where X is going to repress Y, and X is going to titrate Y at the same time. So clearly you're going to stay in a state where you have much X and very few Y. Now, if you have much Y, it's a bit more subtle what is happening, because if you have much Y, Y doesn't regulate the production of X, but Y, it can interact with X. And so if you have, really, really much Y in the system, it can titrate X, so it's going to prevent X to repress Y, so that you're going to lock into a system where you have much I and very few X. So this kind of reasoning that I've just made, like the sound wavy reasoning, you can actually make it more quantitative and really show exactly, identify the parameter range for which you have bistability. And I come back to that. So this is for so this would be bistable with a negative interaction. Oh yeah, and so I should tell you, are there examples of this bistability in actual networks? There is. And actually, if you look at, so this is the question I asked yesterday night. If you look at actually in the lac operon, you have something like this. So in the lac operon, what you have is that X will be, so X will be actually the lac repressor itself. Okay, and so the lac repressor is repressing the lac operon, and one of the products of the lac operon, Y, is an enzyme that is called permease. Okay, and so what the permease is doing is that it allows the lactose outside of the cell to come inside the cell, and then the lactose then titrates the lac repressor. So it's the permease that is allowing the lactose to come inside the cell. And then the lactose here, it titrates the lac repressor. So it's not exactly this network. You have a step here which is via the permease and the allolactose, but that's the exact same topology, the exact same idea that you titrate a repressor to make a bistable system. And the lac operon, so in this context, it has been shown to be indeed bistable. So you have if you activate the permease, if you activate the lac operon strongly, you're going to, and if you have, even with a low concentration of lactose, you are able to fight against the lac repressor and to have heterogeneity in the population, very much like what was discussed yesterday night by Paul, where you have some cells that maintain the lac operon active and some that maintain it inactive based on the past history of the cell. And it can even be transmitted to the daughter cells. So the cell divide and they transmit the state to the daughter cell. So, yeah, so that's bistable. So now, what is the last function that you can have with this mixed feedback loop? And so now, if you have an activation, now if you have X activates Y, and then both titrate. Okay, so now what happens? So now let's try to imagine what very intuitively, again, hand waving, what could happen. So, and let's assume again, and you see where I'm going. Let's assume that the activation X activates Y takes a bit of time. You know, maybe for instance there is a transcriptional step or something like this, which makes perfect sense. You know, there is a transcriptional step, but so let's assume it's not instantaneous. So now imagine you have X that is present. X is present. Okay, so actually let's draw it. Let's draw it because that's going to be even easier to imagine what is happening as a function of time. So X is blue, I think, and Y is green. So let's imagine you have some X. Okay, and so X is activating Y, so Y is turning on. Okay, but then what is happening is that Y is titrating X. So as Y is turning on, X is going to start decreasing. Okay. And so now Y is activated by X, but then since X is decreasing, Y starts decreasing, okay? And then if Y is decreasing, it's no longer able to titrate X, so X is going to turn on again. And then you see what is happening. This is going on for some time. Okay, this guy is actually a nice oscillator. So this mixed feedback loop, like this, can behave as an oscillator. Again, there are very important examples in nature of this. And one very famous example is the Nobel Prize in Physiology and Medicine this year is the circadian clock. Have you heard about the circadian clock? So, circadian clock. So what are the circadian clocks? For those of you who are not familiar with this, so in every single cell of your body, there is an actual oscillator. Actually, there are several of them, but there is a core oscillator that is oscillating with a 24-hour period. And so you get, sorry, you get oscillation in the concentration of proteins, very much like this X and Y. And so, for instance, in fly, because the Nobel Prize was for fly. Okay, so the equivalent of X would be two proteins actually called Per and Tim. Oh, no, sorry, these are the other ones. This is sick and I think it's clock. Should check. Yes, it's at sick and clock. Okay, so this is the protein X. Okay, and so this protein activates other proteins. So the proteins activated are called Per and Tim. Okay, so period and timeless. So that plays the role of Y. The Nobel Prize. So Mike Young, who got the Nobel Prize, is the one who I think discovered Tim and clock, I think. I don't know exactly who got the Nobel Prize for what, but certainly the guys who got the Nobel Prize, they are the guys who discovered these genes. And so now it turns out that then what happens is that per team is produced by second clock, per team is produced in the cytoplasm, and then it goes into the nucleus and it interacts with second clock to relieve the activation. So they exactly do this network here, and this is what generates the 24-hour cycle in the fly cells. And what's very impressive is that this structure of this mixed feedback loop of an activator and then a titration thing. So in terms of evolution, because we always should keep evolution in mind, that this structure of networks is actually very ubiquitous in different organisms. So in mammals, you have the exact same structure, but they are not the same gene. So X, I mean, not exactly the same gene. So in X is a protein called BMAL, and you still have the clock. So we X. And what is per, the role of per team? So you still have per, but you have another protein that is called cry, like cryptochrome. Cry cryptochrome, and it's important protein because, as you can see from the name, it's the one that is detecting the change of light. So this is how you couple your internal oscillator to the change of outside line, because cryptochrome is detecting the change of colors, essentially. It's something that is sensitive to the blue color. And even, so that's for fly mammals, even for fungi, so for instance, Neosporin, sorry, X are proteins called WCC1 and 2. And then, so it's white color, something, I forgot what the last C is, and Y, this is a protein called freak, like frequency. And what is very interesting in fungi that you see, so in mammals and fly, you have some common players. You have clock and per. So you could say, well, they probably derive from the same ancestor. But in Neospora, these are completely different proteins, freak and WCC. So this means that this structure, this mixed feedback loop oscillator that you see in circadian clocks was clearly selected. The structure of the network has been selected specifically for this. I don't think we quite understand that. I don't know why this is the same network structure, but it is. This is an observation that you had some kind of conversion, evolution conversion on the structure of the circadian clocks to be mixed feedback loops. Okay, and so, yes?
You talk about oscillators. This is how it ideally should be, right? So can a lot of disease and pathology be attributed to when this goes out of whack, like they're not in sync?
Are there pathologies due to circadian clock problems?
Like there's a whole range of diseases called.
There's a lot of there are many problems. If you have problems with circadian clock genes, you're really in trouble.
Right. Like prion diseases or.
I don't know about prion disease, but for instance, like
insomnia.
People having mutation on BMAL can have very bad, cancer and stuff like that. And it's also, more practically, people who are working at night, it's well known that they have disease problem because their circadian clocks are essentially completely out of phase with the outside world. So, there is a lot of very current literature on the connections between circadian clock and disease. It's kind of an expanding field. It's also an expanding field because there is money to do, because people want to I'm coming from Canada, I'm really jet lagged. If I had a pill, that's why I'm speaking too fast, probably. I'm sorry. I'm like, I need coffee, No, actually, no, I'm always speaking very fast. That's but anyway, so there's a lot of money and research these days on circadian clocks and pathologies. Okay, I'm going to tell you a word about my contribution to this field. And why is it close to my heart? It's actually, you can do some calculation on this. We were the one who, I think, proposed the first that this was bistable and oscillating, and we were the one who studied that. And the interesting thing is that you can sorry about that. You can try to draw a kind of phase diagram to see in which parameter range you have a bistability and oscillator, very much like what you do in physics. And the phase diagram of the mixed feedback loop is actually very, very simple, very interesting. So I just draw it very, very, very quickly. so this is work I've done with Vincent Hakim during my PhD. And so it's a PRE paper. So basically, you have a simple phase diagram for the dynamics of the MFL. You have two parameters. So let me call them rho 0 and rho 1. And I can rescale them to be equal to 1. And basically, you can draw a phase diagram. So I draw the phase diagram first, and then I tell you what this parameter corresponds to, where here you are in the Bistable regime. Here you have in this oscillating regime. Okay, and so here I should be careful. You are in monostable, but Y is high. And here you have monostable, Y is low. Okay, so it only depends essentially on two parameters. And these two parameters that I call rho 0 and rho 1, they correspond to so rho 0 is basically the ratio of the maximum production rate of Y of X. Oh God, I've confused X and Y. Yeah, I think it's Y. Of Y versus X when so that's when X is does not regulate Y, and rho 1 is the same when X regulates Y. Okay, and so what that means is that, what that basically means is that if you are above here, you have a repression of Y by X, but the strength of the repression is, so the reason why you have a diagram like this is because really what happens, so it's not only a matter of repression, it's a matter of stoichiometry of X versus Y to make the dimerization. And so that's why that even, so if you're here, you basically have a repression of Y by X, because it means that if you're above here, it means that you have more X when we have more Y than when X is not regulating. So that's why you have a repression. So this part here is repression, and this part here is activation. Okay, and so the 1,1 is exactly when you have exact stoichiometry in terms of production rate and dimerization. So this diagram can be intuitively understood. And this diagram is true in the limit where you have very, very fast interaction of X and Y. But anyway, so you get a very simple diagram. It's not so easy to compute the actual line. So it's an idealization. This is in the limit where gamma, where the parameter that I call gamma is very, very big, accounting for the interaction. What happens if you have something that is not instantaneous? It's more like something like that. And so you lose a critical point in the middle if you have if you don't have an instantaneous interaction. So this calculation are not that easy to do actually, but anyway, it's just to show you that you can do something a bit like physics even on this network computing phase diagram. I like phase diagram. You'll see another one tomorrow. Okay. Okay, any question on that? We can move to point 2. Don't worry, my point 2 and 3 are more shorter than point 1. So you should not be concerned that I'm not going to keep you too long. And you have 3 hours with me tomorrow anyway, so. Okay, so. Okay, so so I want to tell you a bit about synthetic biology. So why is synthetic biology interesting in this context? It's because for 2 reasons. So the first reason is that clearly these small modules seem to have some functional. Yeah, you have a question? Yeah. Yeah.
And in the experiment, maybe I was able to calculate and monitor the enzyme activity, different enzymes in a particular system.
Yeah.
So at the end of the day, I have different enzyme activities.
Yeah.
And I've been able to put them. Can I use the activity to model the network to know the particular?
Let's have the discussion tomorrow.
Okay.
Okay, I postponed several things tomorrow, but it's because the question you asked are much more relevant for what I'll be telling tomorrow than this is more like the introduction for tomorrow, actually. So that's why I answer this question tomorrow. Quick answer is yes in some context, but that's not really helpful, but you will see what I mean tomorrow. Essentially, the goal tomorrow will be to reverse engineer network motifs based on consideration like this. Like if I give, if I have some phenotype, can I infer something about the network performing this phenotype? This is what we'll do exactly tomorrow. Okay? Yeah. Any other question? Okay, so synthetic biology. So it's interesting because so the idea that motifs and small modules are important for molecular biology, that seems obvious, but that is not so obvious. It could have been some kind of weird stat-mech things happening in the cell that would not be so easy to engineer. This is all. I don't know if you read What is Life by Schrödinger, but this is a central point of the discussion of this book. Is this that it's not stat-mech happening inside the cell. It's really like small number of molecules that can perform function. And even though this is physics, this is more like nonlinear physics than stat-mech, basically. So I guess we can have a philosophical discussion about that, but so people realize that these modules are important, and so I should have a Feynman quote, because you should always have a Feynman quote in any series of lectures. Feynman said, "What I cannot build, I cannot understand," and so people started to actually build this kind of small modules and see how they're behaving inside the cell, and in a way, checking that this module vision, this idea that a couple of genes interacting could have interesting dynamics, is correct. Okay, and so I'm going to mention two of these works, which are kind of the founding papers of modern synthetic biology. And interestingly, there are two back-to-back papers in Nature in 2000. And so if you haven't read them, I really recommend that you have a look at them, because they really studied the field, an entire field, and I think they're still quite relevant and interesting. And not surprisingly, what this paper did is that they actually built, it's coming back, a bistable and an oscillator using small networks like this. And so, but what is interesting is that the networks that they built are quite different than the one I actually showed you, which also tells you something, I think. So the two papers in question, so this is Nature 2000. Okay, so the bistable system. So it's a group of Jim Collins. And I don't remember who the first author is. That's very embarrassing. I think it's, you remember? Garner? I think it's Garner. It's Garner et al. Garner, Cantor, Collins, I think. 2000. So the bistable system they built is something called the toggle switch. Okay, so what did they do? They did it in E. coli. So they built a plasmid where they encoded two genes, and the name of the gene is not relevant. Let's call that X and Y. Okay, and so what they did to do a bistable system is that, so I told you yesterday a way to do bistability is to have a positive feedback loop. They did a positive feedback loop, but via an intermediate step, which is Y. And so the way that they've done it is that they say that X replacing Y and Y is replacing X. And so overall, it's a positive feedback loop, following the rule that minus times minus is equal to plus. But it's not a positive feedback loop the way we saw yesterday, where it's a self-activation. It's more like a mutual repression. Thus the name toggle switch, because it's if X is high, Y is low, is Y is high, X and low, so it's like switching between an on-off position. And then the way to control the switch, what they've done is that they had other chemicals. One is IPTG, and that's a chemical, and the other one is temperature, where they could modulate the strength of this repression. And with this modulation, they can flip the state of the system. So for instance, imagine you are in a state where X is Y, then what you can do is that X is I, repressing Y, then you put some IPTG. So what it's going to do is that it's going to relieve this repression, so Y is going to turn on, and then Y is turning on is going to repress X. So that when you remove the IPTG, you will stay in the state where Y is high and X is low. So this is a way that you could toggle this thing. And so they've done a construct, and they've done all the security checks that you could imagine. They check that you could switch the state of the system, so the state of the system was persisting when the signal were removed, where the IPTG and the temperature were removed, and so on and so forth. So they've done that. So what I find interesting is that again, this is a small module, but this module is not, it's not part of the module I've told you about. This is a main design module, and it's not entirely clear that in this module, I mean, as far as I can see from Alon's work, it's not overrepresented in the E. coli network. I mean, you find it in other contexts. I'm not saying that it does not exist, but you see, how we design compared to evolution. It's a symmetrical thing. It's there is some aesthetic thing that we see here. And if I show you the mixed feedback loop, I showed you the mixed feedback loop earlier today. It's an ugly design. It's completely asymmetrical. One is via transcription, the other is via protein-protein interactions. So there might be a lesson here. I mean, I'm not going to elaborate too much on that, but I think there is something it's interesting to see that we design things differently, maybe than nature. So that's the first bistable system in synthetic biology, in this very famous nature issue. The second one is an oscillator. Okay, and so this is a paper by Elourty and Leibler. Okay, so 2000 is the same issue. So they're literally back-to-back papers. You can print the 3 pages, and you have all the papers. It's beautiful. Of course, you no longer print papers now, but when I was a student, I did that. And so Elourty and Leibler, they designed an oscillator called the Oscillator. And this is something you've seen already with Jeff Gore. This is this famous rock, paper, scissors system. So what they did, they had 3 genes, so let's call it ABC. And what they did is that they built, a global negative feedback loop with three repressions. So they built this synthetically, and they put that in E. coli, and they have a GFP reporter, and they see beautiful oscillation of the oscillator. So it's a rock, paper, scissors oscillator. Okay. Have you seen why this oscillates, or do I need to know? Is it okay? Is it clear for everybody how this thing oscillates? And it's pretty. Okay, I can do my kind of little drawing again. So. So for instance, let's start with A. So if A is I, okay, so A is I, so A represses B. So if B is repressed, C can turn on. So you have A, then C is turning. So when A is I, C is turning on. And then since C is repressing A, then A is dying. Okay, so you start with C. And then now when C is I, so C is I is repressing A, so if A is repressed, B can turn on, and so you get B turning on. Okay, and that's it. And then you have your cycle. When B is on, A can accumulate, and so on and so forth. Okay, so that's going to look like that. Okay, so that's my oscillator as a function of that. So that's my oscillator. So you see the direction is ABC, but the time is more like BA, whatever. That's not That's not difficult. And of course, you can do all calculations you want, compute the bifurcation, blah, blah, blah. Okay, so what also is interesting is that, again, this is an oscillator, but this doesn't look like any kind of oscillators I've told you about. I mean, you could argue that having, A replacing B, replacing C, replacing A is like a bit like a delay. But I think it's a bit different. The philosophy is clearly different because it's not like you have only one negative feedback loop. You really have, it's a global negative feedback loop, but it's via 3 repression, which is a bit, it's a bit different. It's a bit more complicated. But, that's, so people are still very active on this. So people have, for instance, there was a paper last year. Actually, no, it's not. It's no longer in Elourt's lab. So that's somebody who with Elourt's. So a guy called Laurent Pauvin-Trottier, who was with Johan Paulson and who improved the oscillator to make it much more robust and much more resistant to fluctuation. So people are still, working on the design of this network, and they're learning good biology from it. It's still a very active field, and it's still quite difficult actually to, put these networks, put them in cells, make them work. It's still very, it's tough. It's really tough. That's what we can tell. And even so, I don't know if with people do CRISPR stuff like this, if it's much easier. Actually, I don't know. Okay, so what else I want to say?
Paul.
Any question on that? Yes.
Yes. So I think one of the take-home messages of this school is that rock, paper, scissors is what humans imagine,
absolutely,
but nature never does.
Yeah.
So, have they found these type of motifs in E. coli networks?
Okay, no. As far as I know, there is no rock, paper, scissors transcriptional networks in anything I'm aware of. So I think the take-home message you gave is perfect. It's exactly this. The way nature is thinking is not the way of thinking. It's not the way we would think a priori. We are, we are engineers. We like symmetries. We are physicists. We like symmetries. Nature does not care. Nature is just doing what is efficient, or what is easy to evolve, or something like that. And so that's why, so tomorrow I tell you about evolution in silico, you will see, I think that's why you need to somehow simulate these things in the computer to see, if you have a mathematical model of evolution, what kind of networks would you get? I think that's why it's important to do this exercise and not to be too biased about the way you do it. Otherwise you will find...
Right. So, so it opens many questions, right? Not knowing that nature does not this. One of these is, why doesn't it never evolve? I think you're going to talk tomorrow.
Yeah.
And the other question, which is very much similar in population biology, is how is it able to sustain so many genes?
Yeah.
If you are limited in this type of restrictions, let's say, it's kind of the question of biodiversity, right? How do you manage to sustain so many genes at the same time if you easily get extinction, if you easily, lose control?
Yeah, but first, I don't know if it's exactly the same question, but I'm not 100% convinced that there are so many genes that we sustain. Like, if you look at the size of our genome in terms of number of genes, I would argue it's quite small. If you think about all the diversity of biological behavior happening in our body, we have I don't know how many genes we have, something like a couple of thousands. I would say it's, how many genes do we have? Somebody should know that. I should know that. How many? 20,000, yeah. But I don't think it's much, coding, I would argue it's not that much. But the thing I want to say is that you might not need many genes, because I think one take-home message I wanted to give you. So these are networks with 1, 2, 3 genes. They are very simple networks. But you saw, for instance, the feed-forward loop. Just the feed-forward loop, I showed you 3 completely different dynamical behaviors that you could have with one single network with 3 proteins. And so, essentially adding connections to this, even with this very small network, can generate, huge complexity in terms of dynamical behavior. And so you might not need that many genes to do, and then of course there's some kind of combinatorics happening. If you have two feed-forward loops together, clearly, in parallel, the way they talk to each other, they can generate many, many, many interesting behaviors. So I think the complexity is here. It's not so much the number of the genes, it's in the way they interact. And clearly, one of the take-home messages is that even very small networks with small number of genes and small number of interactions can have very rich behavior. I mean, I spent 3 hours telling you about these behaviors. It's not because I'm not slow, it's because there are really interesting things to tell about these things. So, okay, so 10 more minutes. Okay, so, I don't think I use my 10 minutes. It's really a word. It's a word on modularity. So, what do I mean? What is modularity? First, what is modularity? So, this thing, of course, this is synthetic biology, but I've told you about network motifs, and so sometimes we call them network modules. And so, as I just told you, these modules can have some kind of rich dynamical behavior. But of course, you might imagine that, why would you need to keep these small things on, why would they be independent? You could get, if I want to do an oscillator, I could have many genes talking together and making a huge oscillator where everything is oscillating. And in some sense, this is also what is happening, because if you look at the genes, for instance, all the genes that are regulated by circadian clock, you see many, many, many genes oscillating. Really a significant fraction of the genes in your body and the proteins of your body are oscillating with the circadian period. And in other contexts, tomorrow I will tell you about another kind of oscillator, a developmental oscillator. And this is a case where they see many, many proteins oscillating, but they're completely unable to identify what is a core pacemaker. So this is a question people ask. So why is it that you have small modules performing function, potentially, versus a kind of more like global aspect, or StatMech thing, like some kind of emergent behaviors of all the interact. And so I think we don't have a good answer to this question. I give you the one answer, which is again, Alon proposed this in a paper. And this is an instance where I think I disagree with him, and I tell you why tomorrow, but let's see what his hypothesis is. So why modularity? Why do you do this module? Okay, and so the idea, so now imagine, of course, you have several functions, biochemical functions happening in your body. You have circadian clock, you might have differentiation genes, also positive feedback loop. And so the idea is that, so let's write in a very, very crude way, let's write the function you can have. So let's say f is going to be a function, and I'm not defining, I'm not doing something mathematically sophisticated. Let's call f and g two functions I have in my body, And so the idea is that, well, I could do something. I might want to do something sometimes in evolution. I might want to, it might be favorable to do f and g, like at the same time. Let's assume it's a Boolean function, and I want to do this. And so now what I could do, I could evolve a network performing this f and g function, and then I might have a very complicated function. I might have a very complicated network. So evolution might select for a complicated network. And so they've done this exactly in one of their papers. They took a relatively complicated Boolean function that could be decomposed into this f and g, and then they gave it to the computer. And they try to optimize the network doing it. A bit like, think about machine learning. It was machine learning. It was not exactly machine learning, but that's the idea. It could give you, and then you could generate a network doing this. And so typically you will find the complicated things. Again, in machine learning, if you're familiar with deep learning, usually you cannot really identify in this kind of black box model. You cannot really understand what it's doing. So they argue that if you do this, you will find a complicated solution. And they make the following remark. So now imagine that maybe at this point in evolution you do F and G, but maybe earlier or at a different period in evolution you might want to do F or G. Okay, so that like it's for some evolutionary time you do this. You do this, and then you change the environment. The climate is changing. A volcano is bursting, and I don't know. Like you do this, something is happening. Environment changes, and then you want to do that. You want to select for this. You select for this. So then they argued that imagine now that in evolution there are, sequence epochs, as we say, where sometimes a cell wants to do F or G, and sometimes the cells want to do F and G. And, and this time could be long time, 4 generations potentially. But imagine you want to alternate between the two. Then they argue that if you want to alternate between the two, the good strategy in terms of evolution is to evolve F and G independently. Okay. And then to connect them with essentially links that are easy to flip. So that, in the period 1, when you do F or G, so you have F and G, you do F and G, and then suddenly you are in a period when you want to do F or G, and so you just flip a couple of, links into your network, and then suddenly you have F or G that is working very well, and then you don't need to re-evolve a big network doing this. And so they argue that if you do this kind of evolutionary game where you alternate between different selective pressures, but so that the selective pressure have something in common, you will evolve some modules of the selective pressure that will allow you to basically evolve more rapidly. And so they argue that this is the reason why you have modularity in this gene network. It's because essentially you have selective pressures that are changing in time, but there are some kind of constant in the selective pressure that might be modulated differently. And these are these constants that evolution has learned to optimize into some modules, and then other things are just optimized very rapidly when the environment is changing. So that's what they argued as the origin of the modularity. So I think it's an interesting hypothesis. Oh yeah, I should say they've done some simulations to show that on very simple Boolean examples, where they look at different functions and they do this exact evolutionary game, and they see indeed that modularity is evolving. So I think it's a very interesting hypothesis. I'm not entirely convinced that this is correct. I explain you why tomorrow with my own evolutionary simulation. And with this, I'm going to wrap up. Yeah. Yeah, I agree. Yeah. That's true. I entirely agree with you. So I don't know if you've heard. So the remark is that Alon's networks, they look modular, but if you look at Eric Davidson, who is a very famous developmental biologist who drew these big networks, also they do not look modular at all. So it's true. That said, Davidson is still identifying some modules corresponding to different steps of development. So I don't know how much, maybe it doesn't. Maybe there is some hidden modularity. But you're right; it's not entirely clear that these things are actually modular. But I think it's part of the debate. Is biology modular? Is it evolving to be modular? I don't know. I mean, and can you measure modularity? I don't think you can measure modularity in a simple topological way, I don't think I think it's more like in terms of dynamical systems, like, number of bifurcation or stuff like that. Maybe you can do something like this. But, number of attractors. Yeah. 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 Yeah, that doesn't look more, But the related question is that can you coarse grain these things too? Because maybe if you were able to coarse grain them in a clever way, maybe you will be able to identify better if there is some modularity and quantify it. Okay. Any other question? Okay, thank you.